Welcome everyone to the Education Day session on genetic testing presented by the ISPG Genetic Testing Committee. I'm Francis McMahon, chair of the committee, and on behalf of our committee members and my co-chair, Francisca Degenhart, I'd like to welcome you to today's presentation. We have an exciting lineup of speakers who will give a quick overview of a broad range of topics related to the use of genetic information in clinical psychiatry. We also will be followed by a half-hour session where our participants can join chat rooms for what we hope will be a fairly informal and broad-ranging discussion on a whole range of topics related to the use of genetic information in psychiatry. See if I can get my slide to advance here. There we go. So the ISPG Genetic Testing Committee, if you're not familiar with us, we're an interdisciplinary group of preclinical and clinical researchers who are drawn to represent a range of fields, genetics, neuroscience, psychosocial research, ethics, psychiatry in general, psychology, medical genetics, genetic counseling, as well as genetic testing laboratories. And we're fortunate to have representatives from all of those fields on the committee, some of whom you'll be hearing from today. We have a consensus statement that we generate every couple forms of genetic information in their clinical practice. And if you are not familiar with it, I can refer you to the most recent consensus statement, which is available online at this address on the ISPG website. Our charge is to periodically review the available evidence and formulate recommendations aimed at providing guidance for clinicians on the use of genetic information in clinical psychiatry. So today we'd like to bring you up to date on where we are with our current thinking on these topics. And I'm going to turn over to my co-chair to bring you through the outline of today's presentation. Francisca?
So today you will receive an update on what kind of role genetic information plays in the diagnosis and treatment of common psychiatric disorders, with both looking at what currently is being offered and also with an outlook into the future. Then we'll move on to what clinicians and patients need to know about genetics in order to fully understand genetic testing and its implications. We will take a look at the key ethical and social issues relevant to these topics, and then how genetic information is best communicated so it's fully understood by patients and their relatives. This is an outline of the 6 speakers that we have. I won't go into detail to save some time. And this is just a quick reminder that after the presentations that we will continue the discussion in breakout rooms, and that there will be separate breakout rooms on different topics. So there will be one looking in closer detail or discussing in closer detail diagnostic testing, CNVs, and polygenic risk scores. There will be one focusing on genetic counseling, secondary incidental findings, a third one on education, how to best train everybody involved in genetic testing and genetic counseling, and a fourth one on pharmacogenomics. So thank you very much for everyone who joined. And then we will start with a first presentation by Brenda.
I'm going to be talking today just briefly overviewing diagnostic genetic testing for neuropsychiatric disorders. and really talking about where we are currently in terms of clinically used tests. And I want to start by just differentiating different types of genetic tests that are used in psychiatry. This should be familiar to all of you, but maybe you haven't thought about them in these different buckets. As a genetic counselor and from the genetics point of view, the technologies used for these different types of tests are quite different from each other, and the types of reports we get out of them are quite different. And so it's important to kind of tease them out. I do think in the clinician world, people have a lot of confusion about genetic testing and what is being used for what. So clinical diagnostic testing, which I'll talk about today, is really aimed at identifying very rare causative genomic variants. These have large effects on brain function, and we use these and have used these historically for the etiological evaluation of individuals with symptoms. They've been widely used in pediatrics, and we're starting to see them get used for neuropsychiatric disorders in adults. They're highly penetrant and have a wide range of expression. Of course, these are different from GWAS studies, which are a research tool, and GWAS really, by contrast, is looking at these very common genomic variants that have very, very tiny effects, but they're additive. And, hopefully in the not too distant future, as we'll hear in this meeting, there's the potential for clinical application using polygenic risk scores, but these are quite different from the diagnostic tests of rare variants. And then, of course, there's pharmacogenetics that's looking for variants that are expressed primarily in the liver, but looking at medication response. So today, I'm just going to quickly run through where we are with clinical diagnostic testing. And as I mentioned, we have decades of experience using this type of testing, literally. We had tools in the past such as karyotyping, which, were not particularly powerful, but we've been doing them. And as the clinically available tests have really increased in their power to detect things, we continue as genetic counselors to use these. And these have been for decades the standard of care for children who have autism, developmental delay, intellectual disability, or congenital anomalies. And really the aim is to identify those rare conditions, genetic conditions that might be causative of their symptoms. Interestingly, although this has been primarily, when it comes to neuropsychiatric disorders, a pediatric enterprise, we've had many decades of experience also testing adults sort of secondarily, because many children who get diagnosed with, let's say, a copy number variant or a rare genetic cause of their autism or intellectual disability, we will often recommend parental testing, and then not infrequently we find the same copy number variant in a parent and then provide genetic counseling as well. So it's not uncommon, even in what we've been doing to date, for parents to learn of their own genetic diagnosis, potentially associated with a number of neuropsychiatric symptoms based on a child's workup. So again, the recommendations that have been out there for quite a long time have been primarily based on pediatric experience and have been recommended for children. We recently, at least our group at Geisinger and some others, collaborators around the world have been talking about, this artificial cutoff with pediatrics, that, this can also be applied to adults who have intellectual disability or autism of unknown cause. But the three main tests in our armamentarium, if you will, include a test called FMR1 DNA analysis, which will look for Fragile X associated disorders that can't be picked up on microarray analysis or exome sequencing. And then, of course, chromosomal microarray and whole exome sequencing most recently. And so there are recommendations out there by experts and professional societies that children with global developmental delay or intellectual disability or autism spectrum disorder of unknown cause should have this testing. We do know that it's under implemented even in the pediatric world. So the Fragile X test is one that will look for expansions that are responsible for what we call Fragile X associated disorders. It's particularly relevant for patients who have developmental delays or autism because it's a significant cause of those symptoms. It's a relatively inexpensive test in the States, generally under $100, for example, and you do it once, and if they don't have a fragile suspension, you don't need to do this again, but it has very important implications for extended family, which is why it's recommended. Fragile X is really not something that occurs out of the blue, so once you find it in a child or a person with a disability, there are often many, many other family members for whom this has implications. Chromosomal microarray analysis is a much more powerful tool that looks at many different conditions, not just one condition, and it's really aimed at detecting copy number variants, or what we call CNVs. These are deletions and duplications of genetic material. Most often these affect multiple genes. And, you know, in psychiatry particularly, we tend to point to the example of the 22q11 1.2 deletion, which is well known and well studied and has, in addition to developmental implications and physical implications, because there are a number of medical problems that can be associated with it. We also know that individuals with the 22q11 1.2 deletion have, you know, a 25 to 30% risk of developing schizophrenia. So this is one that's very recognizable to many psychiatrists. However, it's important to put this into the context that there are hundreds of known rare pathogenic copy number variants in addition to 22q, and typically they also have cognitive and neuropsychiatric symptoms, and some have medical comorbidities as well. So chromosomal microarray analysis diagnostically will pick those types of things up. And then much more recently is exome sequencing, or sometimes called whole exome sequencing. And this is an even more powerful test that's really looking for variations in the protein coding regions of the genome. And it's looking at sequence level variants. And again, when we're looking at this level and using exome sequencing as a clinical diagnostic test, we're trying to identify rare pathogenic sequence variants that have very large effects on brain function. And there are, again, hundreds of well-established rare genetic etiologies of neuropsychiatric disorders. This is starting to be, at least in some countries, in widespread clinical use in pediatric populations. It's a very powerful test. And recently, this test has the capability of also picking up those copy number variants and would eliminate the need for microarray in many cases. So that basically now you're down to, you know, Fragile X analysis when indicated and an exome sequence, which will do the work of microarray in most cases. If we look at the diagnostic yields for various disorders, you know, when we talk about neuropsychiatric disorders, there are various definitions. Here I'll use sort of the broader definition, including things like epilepsy, intellectual disability, autism, etc. And you can see here the various diagnostic yields of both CNVs and single gene disorders. So I think we can say overall there's a relatively high diagnostic yield of exome sequencing with CNV calling in individuals who have neuropsychiatric disorders. Well, why are these tests important? And I think it's important to talk about the benefits of genetic diagnosis. And from a medical standpoint, we know that it establishes a primary etiology of clinical diagnoses. Again, these are rare variants that have very large effects on brain function and can be considered causative. This allows us to do some anticipatory medical guidance for conditions where we know a lot about some of the associated medical issues. And then we can also invite families to participate in targeted research opportunities and clinical trials, many of which are going on now for various specific genetic disorders. Families also can benefit from genetic and reproductive counseling about their families, having future children, questions they may have about chances for having these CNVs or other genetic disorders happen in the future. And then also very important, and there's more coming out about this, is the personal utility, even for adults. You know, these families of children, but also for adults, they have access to etiology-specific advocacy organizations, and there are hundreds of genetic support organizations like this. I will just wrap up by saying, you know, there are ways in which medicalizing a condition can potentially reduce stigma, which is a big issue, of course, with mental health conditions, and we have increased empowerment engagement with behavioral health providers. And I will stop there because I don't want to overstay my 10 minutes, and I'm happy to take some questions in the next session. Thank you.
Thanks, Brenda. That was terrific, and I'm sure there'll be a lively discussion at the end on this subject matter. I want to introduce our second speaker, Eva Schulte from the University of Munich, who will be talking about GWAS hits and polygenic scores in clinical genetic testing, a tale of the future.
Well, thank you, Francis. And it is my great pleasure to be able to tell you a little bit about the role of GWAS hits and polygenic scores in clinical genetic testing. And as Francis said, it's more of a tale of the future than it is really the current reality. I'm sure most of you know this, but most psychiatric disorders are common complex disorders with a polygenic genetic architecture. And over the past decades, GWAS, although they require very large sample sizes, have been very successful in identifying common variants that are associated with psychiatric disorders, each variant with a very, very small effect on the phenotype. So then you can use polygenic risk scores to capture more of the heritability that is conferred by common variants by adding up all these very small effect sizes. With this, people then asked whether genetic information from common variants can also be used in a clinical setting. But first, we do need to remind ourselves what polygenic risk scores really are. They are derived from genome-wide association studies and are the weighted sum of the number of risk alleles an individual carries. So at any given risk locus, you can have either 0, 1, or 2 risk alleles. These are summed up and then weighted by the individual effect sizes. That yields a single score of an individual's genetic load for a given phenotype. The way we currently calculate the polygenic risk scores assumes an additive genetic model and an independence of genetic risk variants. So we don't really account for any gene by gene or gene by environment interactions. And what you end up with is what you can see sort of in the right lower corner of the screen is the normal distribution curve of all the polygenic risk scores distributed in any given population. And then you can pick one individual and see where he or she falls on that normal distribution curve. So can we also use polygenic risk scores in clinical applications? The first evidence of that really stems from cardiovascular diseases, which are the flagships of the utilization of polygenic risk scores in clinical applications. And what the colleagues identified there was that if you only look at individuals at the very end, or the extreme ends of the normal distribution, these individuals actually carry risk increases that are comparable to monogenic forms of disease. But what is important about that is that monogenic forms are usually really rare. So you find them in maybe 1 out of 100,000 individuals, whereas the individuals that are identified as having an increased risk using polygenic risk scores maybe are 1% of the entire population. So many more individuals can be identified that have this comparatively increased risk to the others. We also see the situation in psychiatric disorders where, for example, we find odds ratios when we're comparing the highest to the lowest decile of polygenic risk scores that are approximately 3-fold increased in, for example, major depressive disorder and roughly 10-fold increased in schizophrenia. In psychiatry, PRS may be especially helpful just because we have so few biomarkers in general and also very few clinical predictors. However, there are some very important caveats that need to be addressed when looking at polygenic risk scores in a clinical setting. The first one being that polygenic risk scores work best when the training and the test samples are most similar. And this actually introduces a large ancestry bias in the current data that we have because genome-wide association studies assessed European individuals in 80% of cases, but Europeans only represent 17% of the world's population. So when you then try to use the polygenic risk scores that you derived from Europeans and try to predict whatever phenotype you're interested in, even if you set the prediction accuracy to an artificial 100%, when you try to use that polygenic risk score then in different ancestries, your predictive ability starkly drops to a predictive accuracy of maybe 50% in East Asians and 25% in African populations. So using the polygenic risk scores that we have right now, we really leave out 83% of the entire global population where we can't really use them because they're not predictive. Also, polygenic risk scores only explain little of the variance in liability to psychiatric disorders overall, ranging somewhere between 2.5 and 5%. So they will never be standalone predictors, but they may be very helpful when looked at in conjunction with other biomarkers or other clinical predictors. Also, maybe specifically to psychiatry, we also have to ask ourselves, what sort of modifiable outcomes do we have that really, if we know an individual has an increased risk, what are we going to do differently? This is very different, for example, in cancer, where you would just increase screening frequency and you actually have something that you can do, whereas in psychiatry, this is something that we still need to address. Nonetheless, there are a number of potential use cases which essentially span an individual's entire lifespan, ranging from risk prediction, maybe even at birth, through prodromal stages where an individual may have unspecific symptoms that can, and polygenic risk scores could then be used to maybe predict whether these symptoms will resolve or whether somebody will actually develop a psychiatric diagnosis. And even with individuals with full-blown symptoms of psychiatric disease, sometimes we don't really know what diagnosis category they fall into. Here, PRS could also be helpful. They may also aid in individuals who already have a diagnosis to better assess what treatment or management strategies to use, or to predict outcomes, maybe by assessing whether an individual is more likely to have a large number of episodes throughout their lifetime or maybe not. So with that, I'd like to take you through two use cases. The first one being risk prediction and the ultimate goal of prevention. So we are already very good at predicting disease status in research-based case control studies and large population-based studies, and even in real-world data from electronic health records. However, what we really want to do is, or want to see, is whether we're able to predict whether an individual will develop disease, ideally before the onset of symptoms, or in a situation where we don't have a lot of information, where we don't have this 20-year history and we already know that an individual has a given disease. But in order to use polygenic risk scores in that setting, we really need clinical trials, which assess whether polygenic risk scores are able to prospectively predict disease manifestation in sort of a longitudinal assessment. And as long as we don't really have these, there's currently no clinical application for polygenic risk scores in risk prediction. The second use case would be in treatment and management. And I think that anybody who's ever worked in clinical psychiatry will know that it can sometimes be very frustrating because we don't know whether an individual will actually respond to the treatment that we administer. And we won't know that for a number of weeks, sometimes even months. And we also don't know which of two drugs to pick or what general treatment to pick sometimes, just because we don't have any very good predictors. Also, we don't have predictors of unwanted side effects. And these are realms where polygenic risk scores could be very helpful. At this point, polygenic risk scores for schizophrenia have already been negatively correlated with response to antipsychotics, both in first episode patients and also with lithium response in bipolar patients. However, we don't really want to correlate it with this polygenic risk score for schizophrenia, but we would much more like to have a polygenic risk score that actually predicts treatment response. And in order to generate those, we really need very large sample sizes of very deeply phenotyped cohorts where we have different drugs that people use, where we have longitudinal information, and we currently also don't have that and are still waiting for that. So as long as we don't have that, currently there's also no clinical application for polygenic risk scores in treatment and management. This is also reflected in the ISPG's genetic testing statement, which states that currently PRS are not to be used to identify high-risk individuals, and they're also not to be used to diagnose psychiatric patients. However, we also acknowledge that this is something that may very well change in the future. So with that, I hope I have been able to show you some of the potential use cases for polygenic risk scores, but there are also a number of issues that still need to address, especially with regard to the ancestry bias that we currently have. But we also really haven't figured out how to return results to individuals and who would return those results and what we would actually do with them, and also how to treat individuals who basically present to clinical practice already with their polygenic risk scores prepared, which they have gotten from direct to consumer testing. So in quotidian clinical practice, there is currently no use of PRS, but there may be in the future. However, what is clear is that polygenic risk scores will never be standalone predictors of common diseases, and that they will always have to be used in conjunction with other clinical predictors or with other potential biomarkers that we might have. With regard to psychiatry, it is important to know that whenever we want to use a polygenic risk score, we must also think about what sort of specific outcome that is going to trigger, just because currently the situation is that simply because I know somebody has an increased risk for schizophrenia, for example, I'm really not going to change very much in my management or my treatment of that individual. So with that, I'd like to thank you for your attention, and maybe if I get to come back in 5 years or 10 years, I will actually be able to tell you a little bit more about the quotidian clinical uses that we have for PRS in clinical psychiatry. Thank you.
Thank you very much, Eva. We are now moving on to the third speaker, Marcella Rietschel, who will give us a concise overview on incidental and secondary findings, which will become of increasing clinical importance when we move with larger scale genetic testing into the clinic, such as exome sequencing or whole genome sequencing.
Thank you very much, Franziska. So secondary or incidental finding is a finding unrelated to the initial indication of an examination or test. Secondary or incidental findings are not new to genetics or to medicine in general. Any medical examination, such as, for example, a radiological or ultrasound examination performed for one purpose, has the potential to unravel unexpected findings. These findings may be benign with no clinical significance or point to a previously undiagnosed medical condition which may require treatment. While in principle, it can be considered beneficial or interesting to learn about hitherto unrecognized conditions, especially if a disease can be prevented, unexpected findings and their consequences may have negative effects, especially if they increase uncertainties, anxiety, and stress. Genome-wide tests such as chromosomal microarray analysis, exome sequencing, and whole genome sequencing have the potential for discovery of secondary and incidental findings. And these findings may be medically actionable and important, or they may be not. With the rapid evolvement of sequencing technologies, uncertainties have risen with respect to standards of the identification and the interpretation of genetic variant. Thus, in order to promote standardized reporting of actionable information from clinical genomic sequencing, the American College of Medical Genetics and Genomics, ACMG, recommends a list of genes from which secondary or incidental findings should be deliberately pursued, and also offers interpretation which variant would qualify as pathogenic, likely pathogenic, of uncertain significance, likely benign, or benign. Findings in these genes are related to health conditions with known medical recommendations for healthcare providers to act upon with their patients. This list is periodically revised by the Secondary Finding Maintenance Working Group, who uses the terminology secondary finding because these genes are intentionally analyzed as opposed to genetic variants found incidentally or accidentally. Clinical diagnostic laboratories performing exome or genome sequencing should report any of these secondary findings back to the referring clinician, who should be responsible for pre- and post-testing counseling and medical follow-up. In their original recommendation from 2030, the ACMG recommended that this should be done even when the findings are unrelated to the primary medical reason for testing and irrespective of the wish of the tested individuals. Not unexpectedly, however, such compulsory genetic testing did not remain undebated, And the updated recommendations include an opt-out option of receiving secondary findings for individuals undergoing clinical genomic sequencing. Just to mention, a recent ACMG policy statement, which is meant to reduce unproven intervention based solely on phenotype information, the ACMG points out that the list of the secondary finding version, these are 59 genes, was not validated for general population screening, and many of these genes have uncertain penetrance when identified in asymptomatic individuals. So what are the implications for clinical genetic testing? Patients who are offered genetic testing should be informed about the possibility of such secondary as well as further incidental findings, and they should be asked whether they wish to be informed in case such a finding is detected. A real autonomous decision, however, can only be made if it is based on knowledge. This implicates, among others, that patients have to be informed about the types and the probability, it is the frequency of possible secondary and incidental findings that may be found. Just to mention, a most recent study where almost 22,000 participants were drawn from a variety of settings. It is primary care, biobank, specialty clinics, which showed an overall frequency of medical actionable secondary findings of 3%, and secondary findings were associated with cancer susceptibility most frequently, followed by cardiovascular diseases and lipid disorders. The question arises, if it's really feasible and possible to adequately and comprehensively inform all patients undergoing genetic testing in advance about the meaning of a large number of potential secondary and incidental findings and about the possible consequences these may have for them and for their families. This requires them to understand if no secondary or incidental finding is identified, this does not guarantee that no genetically based illness will ever occur. This is the easier part. More important and difficult to understand is that if a secondary finding or incidental finding is identified, there may still be interventions that mitigate adverse health outcomes, such as enhanced health screening and more frequent checkups, dietary intervention or specific preventive treatments, which may cause extra cost and stress to the patient. An individual suffering from a mental disorder or caring for a child with a mental disorder prepared for undergoing genetic testing may perhaps not wish to be burdened by more information and potential worries. For example, that a secondary finding in a child may indicate a cancer risk in a parent. This can cause a lot of stress. On the other hand, what about the duty of a physician if a patient does not want to be informed and a pathogenic variant is nevertheless identified? Assuming that there may be some ambivalence in patient, we did a small study asking bipolar patients and their relatives, what is your attitude if my physician is entitled to disregard my wish concerning the disclosure of secondary finding? Not unexpectedly, almost more than 90% strongly disagreed to this statement. But if we added a further sentence saying my physician is entitled to disregard my wishes concerning the disclosure of secondary findings if good treatment options are available, you see that the disagreement almost vanishes and turns into almost an 80% agreement to the statement. And finally, if we ask what weighs more, the physician's duty of care towards me as a patient or my right to decide what I want to know about myself and what I do not want to know about myself, you see that the situation is not clear, and it may be if you will find a pathogenic variant and the patient had said in advance they do not want to know about it, and it turns out that it could have been prevented if they had learned about that these relations may even shift. And this all indicates that genetic counseling is definitely warranted to encounter such problems. And again, there's a question, are there enough personnel knowledgeable in the interpretation of secondary and incidental finding, and at the same time experienced in counseling patients with mental health disorders? And the next speaker will probably answer this question. Thank you very much.
Thank you, Marcella. Really interesting topic, and I'm sure there'll be a lively discussion about that in the chat room at the end of our session. Our next speaker in today's session is Jehannie Austin from the University of British Columbia, who will be talking about psychiatric genetic counseling. Jehannie?
Thanks, Francis. I hope everybody can hear me. Good morning, afternoon, evening, wherever you are. Very nice to be here. So yeah, I'm going to talk about psychiatric genetic counselling for the next couple of minutes. That reminds me to set my timer. Good, okay. Yeah, so let's start with the very basics. What is it? It's not genetic testing. That's what I can tell you straight away. I mean, it's incredibly relevant to the context of genetic testing because one could make the argument that if you're doing genetic testing for somebody in the context of psychiatric disorders, then it's probably a really good idea to make sure there's some degree of genetic counselling that accompanies that. But genetic counselling is about way more than just genetic testing, and that people can benefit from it even without genetic testing being part of the picture. So just to get a bit more of a handle on that, here's the actual definition of what genetic counselling is from the National Society of Genetic Counsellors, which is the largest and oldest professional association for genetic counsellors, of which Brenda and I are actually both ex-presidents. But it's defined as a process of helping people to understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. So as you can see, there's no mention of genetic testing in there. So it's a way bigger umbrella of an activity than simply talking about genetic testing, although that can be a very important part of it. So the informational component of genetic counselling is really about helping people to understand causes of conditions, how that relate, and in the context of psychiatric disorders, how that relates to recovery, reducing risk and chances for other family members to develop a condition, if that's something that the patient is in or family member is interested in talking about. There's a support component, which is absolutely critical, which is about identifying and addressing issues like guilt, shame, blame, fear, stigma, that often attach to people's explanations for cause. So we know that without providing a really concrete, coherent explanation to people about why a psychiatric illness emerged, people will make their own explanations, and these emotions often attach to those self-developed explanations. However, it's super important to provide genetic testing if it's to provide genetic counselling if there's genetic testing being done. But as I've said, super helpful even if it's not. So, given that I just told you in the definition that genetic counselling is about helping people and understand and adapt to the genetic contributions to disease, like what can we actually say to people about the genetic contributions to psychiatric illness, just as a foundational baseline here? Well, what we know in general is that psychiatric disorders arise as a result of the combined contributions of genes and experiences acting together, right? So you heard Brenda talking about CNVs, and CNVs are really interesting examples of situations where you can have a deletion, for example, 22q, which causes a syndrome, but it doesn't necessarily, depending on how you define the word cause, of course, cause schizophrenia, let's say, because only 30% of people with 22q develop schizophrenia. So depending on how you use the word cause, it's, you know, the syndrome itself doesn't cause the neuropsychiatric phenotype. There's something going on. Otherwise, everybody with 22q would have schizophrenia. So in general, what we understand about these conditions, that it's genetic stuff and experiential stuff acting together. And while that may sound really trivial to you or I, that can actually be really impactful and meaningful for families to hear about. I'm just going to share some examples about how. So these are just a bunch of quotes from real people who've participated in studies over the years talking about their perceptions of cause. there's always just this thought that maybe you're just a bad person, you're just lazy. It's so hard to know what you did wrong. The feeling that we somehow caused this is strong. This happens because we're judged harshly due to our child's behaviours. I was lectured by family members about parenting skills. It came from my side. I've got the guilt. If I hadn't had him, he wouldn't be like that. If I'd known more at the time, I probably wouldn't have had any children because of what I've seen happen to him. I didn't think about this being passed on when I was 23 years old. You think this will never happen to me? So all of these quotes from real people exemplify the ways in which understanding cause of illness is critically important to people from a psychological perspective. And this is really what genetic counselling can help with. So I'm just going to share with you a model that we use in the genetic count. We've developed a specialist psychiatric genetic counselling clinic here in Vancouver that's been running for the last 8 years. We've helped over 1,200 people, I think, at this point. And we've got lots of research papers out there talking about the outcomes of the service that we provide. But just to give you a bit of a snapshot about some of the things that we do. So we use an analogy to help people understand how genes and experiences work together, because it's nice to say the words, but it doesn't really actually give you anything tangible to sink your teeth into, and that's what this jar model is about. The concept is that we all have a mental illness jar, and that in order to be actively experiencing an episode of illness, one's jar has to be filled up to the top. And there are 2 different kinds of vulnerability factors that you can use to fill your jar. The orange balls in the jar here represent genetic vulnerability factors, and the blue pyramids are representing environmental vulnerability factors. So we talk with people about how we all have some genetic vulnerability. It stays the same constant over time, but what can change over time is the experiences that we have during our lives. And so, you know, the more genetic vulnerability you have in your jar to start with, perhaps the more likely it is that you will develop a mental illness. The less in your jar to start with, the less likely you are to develop a mental illness, but we all have some. That's what we know from the genome-wide association studies, right? So yeah, that's just reiterating that point. So most of us are just going to be sort of intermediate in that moderate genetic risk range. So really, it just depends what happens to us during the course of our lives that determines whether or not our jar gets full. Now, my favorite thing as a genetic counselor about talking with people about psychiatric disorders is that we don't just get to talk about how people get sick. People recover from active episodes of psychiatric disorders. The label bipolar doesn't go away, but you're not constantly necessarily in a state of mania or depression. So I really enjoy talking with people about how to protect mental health. And so the concept that we use here is that you can use protective factors, which are rings that stack on top of the jar, making it taller, so that it can accommodate more environmental stuff without getting full, essentially. And so evidence-based protective factors, if you like, would include things like sleep, nutrition, exercise, more effective ways of managing stress, social support, and then there are individual things as well. So the genetic counselling is really about having a conversation with someone to understand what they know for themselves helps to decompress their mental state. It might be spending time with a pet, might be you know, a church or a religious group, it might be mindfulness meditation or what have you. So helping people identify what it is that works for them. I just want to very quickly share a case example with you. I mean, you know, it's nice to hear about it in theory, but let's talk about it applied for a sec. So obviously bits and pieces have been changed to protect identity, but this is one of my favorite cases from a while back now, but somebody I always called Bob. He had a diagnosis of schizoaffective disorder, and I provided genetic counselling for him actually in hospital because he was not doing well psychiatrically. He'd actually recently attempted suicide. He was in the hospital fighting with a psychiatrist about medications. The psychiatrist obviously was saying, if you want to get better and you want to get out of here, you're going to have to take this stuff. And Bob was saying, nuh-uh. And there was a whole lot of not much happening, basically, and everybody was quite frustrated. So we had posters up around the hospital that were basically saying, do you have schizophrenia, schizoaffective or bipolar? Would you like to better understand what caused your illness? If so, give us a call, which is why I was there, basically. He'd responded to that. So I sat down and talked with him about, you know, to do the genetic counselling. But before we began, I did a little bit chit chat, get to know you. Turns out Bob actually had a graduate degree in psychiatric genetics. which was interesting. We had a little nerd out session. It was great. But it made me assume that I knew what he would attribute his illness to. I assumed that he would attribute his illness to genetics. But when I asked him, can you tell me what you understand about the cause of your illness? He said, bad life decisions. So I was a bit baffled and said, what do you mean? And he talked about how, you know, this particular decision, another particular decision, the fact that he'd smoked too much cannabis as an adolescent, and he felt that was why he had his specific illness. However, when I took his family history, both of his parents had psychiatric illnesses, not the same one that he did, but very closely related ones. So I drew that out, showed it to him together with the jar model, and he burst into tears, talking about how he could see for the first time that perhaps it wasn't all his fault that he had schizoaffective disorder. He talked about a weight of guilt being lifted. And 1 month later, he was out of the hospital taking his psychotropic medications. And when we followed up with him to ask why, he explained that once he understood that there was a biological component to his own specific illness, then a biological treatment, i.e. medication, started making sense. Okay, so hopefully that illustrates for you how genetic counselling is about more than just helping people understand the benefits and limitations of genetic testing, although it can be incredibly useful for that too. Outcomes quantitatively of genetic counselling include things like increased self-efficacy, increased empowerment, lots more data about that to share with you, but I'm going to skim over that to the summary because I'm out of time. So cancel, stop. Thank you. Understanding cause of illness is profoundly important to families. Yeah, we've discussed that second bullet, so I'm going to leave that. But basically, I think when communicating about genetics and psychiatric disorders, we have to be really thoughtful to ensure positive patient outcomes. And that if we're doing any kind of genetic testing, whether it be CNV testing, PRS testing, etc., we really need to make sure that we're delivering that in the context of an evidence-based intervention, which is really what we have in the context of psychiatric genetic counselling. Thank you very much.
Thank you very much. We'll move on to the next speaker, who will talk about how all the issues that were just outlined to you on genetic testing, but also on genetic counseling, how probably hardly any of us had this in curricula training, so at university or college. So how we will move how we will move to educate everybody involved in mental health on these important issues. So Fernando, please go ahead.
Thank you. Good afternoon. Buonasera to people in Italy. I wish I was there. So I'm going to talk a little bit about the complexities and opportunities really for educating clinicians, right? The people that will ultimately order most of the testing. So let's start. What do clinicians want to know? And in a sense, it's relatively simple. They want to know the diagnosis of a patient. And here, of course, we can potentially help them with diagnostic testing. They desperately also want to know about prognosis. And I call this work in progress because I think this is something where the field needs to go a little bit more. I mean, the studies are just beginning in terms of outcome and prognosis of our disorders. And the other thing is they want to know what treatment is most effective. And here we can potentially, again, be helpful in terms of pharmacogenetics. So that's a way of sort of framing why clinicians should know about genetic testings. The challenge, of course, is that clinicians really want simple and often actionable answers. And you're competing with a lot of other information. And the problem, the problem with the evolving data and the evolving evidence from our field is that it's relatively nuanced and we don't necessarily have very simple and actionable answers. So this is from a very clear, concise, and wonderful review by John Nurnberger and colleagues from this society, but independent of the genetic testing task force. And what I've highlighted here, it's a very well-written paper, but they underline certain points that I've highlighted here. And the issue is it's only really 5 points because the data, again, is nuanced. And when you try and actually describe what is it that we want to, what prescription, what clear message do we want to tell our clinicians, it's relatively few messages. And that's one of the challenges and one of the opportunities, right? So as we've heard from before, I won't repeat this, really it's the empirical risk from family studies. And of course, clinicians sort of know that, so they want more more than that. Chromosomal microarray techniques, again, there's been tremendous advances here. I'll just add one thing that, at least in my hospital, the people that really order the chromosomal microarray techniques are, of course, genetic counselors, but mostly it's pediatrics, developmental pediatrics, and neurology. So far, psychiatry and child psychiatry have not engaged very much in their ordering or in their understanding of microarray techniques. And then the pharmacogenetics, which of course Chad will talk about, but just to say that there's so much pharmacogenetics information out there, but here's just the limited information that we're sort of told makes sense. And one, of course, is the HLA testing for patients of Asian descent. The other one is two very sort of light touch recommendations about pharmacokinetic variants, and two medications. There's many medications out there. This is just two. And then it's a sort of appropriate statement about these pharmacogenetic profiles that are really out there and are flooding the market, at least in the US, but there's caution about these pharmacogenetic profiles. So this is very reasonable. I say that, you know, this is relatively light touch, and it's not because they could have been much stronger, I don't think. Really, it's about the nature of the evidence that it's relatively incomplete right now. And again, this is just their abstracts, and it's just highlighting that usually one of the difficulties in teaching this topic to clinicians is that we tend to make these sorts of statements, right? Genetic testing is recommended, but its use is currently limited. Commercial testing is widely available, but its utility has not been clearly established. Other methods will soon be clinically applicable. So there's a back and forth and nuance that makes it a little bit challenging. So of course, I think we need to go back to the basics, and we need to get clinicians really at the beginning of their medical education. And I'll talk a little bit more about that. So I want to give just two again, examples about the state of the art, the state of the evidence, and why it is appropriately difficult to interpret right now. So even this is sort of our, this is should be one of our easiest genetic tests, right? This is the 22q deletion. It's something that we've known about for a long time. It's our kind of flagship in terms of identification of genetic testings, again, in adult and child psychiatry, not necessarily in the neurodevelopmental field. And here, the nuance is really, so you look at the PGC paper, and it's the largest paper by far, and you look at the effect size. So when we talk about informing clinicians, of course, they don't care about p-value one little bit, right? What they care about is the effect size and what is it associated within a patient population. And so here you see this effect size of 70 almost, right? And you think this is almost Mendelian. Quasi-Mendelian. But really, thankfully, you see that in Denmark, where you do a sort of epidemiological type of study, again, this isn't entirely comparable. One is hazard ratio, one is odds ratio. The effect size is much, much smaller, right? So here is complexity, and when we educate clinicians, we have to think about how to educate this. We can't, as has been said already, this is usually thought of as a schizophrenia risk gene. And really the data has made it much more complicated. It is far more than schizophrenia risk gene. It might not be a primarily schizophrenia risk, actually copy number variant, not a gene, sorry. Here are two examples from the clinic. Now, my clinic is just a general mood clinic, and it's not a specific genetics clinic, but this is two examples that sort of show the problem, the need for education, and the problem of misunderstanding of genetic testing that is the status quo. So one is a complicated patient. He had genetic testing, and in the genetic testing, it said he was a 2D6 extensive metabolizer. And the clinician and both the patients saw the word extensive and immediately assumed that they would need very high doses of medications. And then I see him on these very excessively high doses of antipsychotics and mood stabilizers, and maybe to no one's surprise, he's having concerning side effects, right? Concerning motoric side effects. Here's another one, and this shows you one of the issues of communicating information and how potentially difficult it is. Now, to their credit, some of these commercial platforms have done a nice job of actually showing it and of making it clinician friendly, but sometimes it's just wrong or inappropriate. So this is GeneSight, which is the most widely used. Myriad has taken over. And this was just a 28-year-old, again, person with bipolar disorder, got one of these red boxes telling her that there was something with lamotrigine. Now, the clinician, the patient and the clinician immediately jumped to the conclusion that they should be off lamotrigine. So they stopped lamotrigine. And really, you have to see this little 2 and then read the little 2. And it says serum levels may be too low, higher doses may be required, right? So the issue is you don't have to be off lamotrigine. We're not even sure if you need higher doses. There's a lot of variability in lamotrigine, right? So here's something that really didn't help and it misinformed. Again, it misinformed because clinicians didn't go through all the trouble of reading, but you've got to assume that people are busy and they're not going to read all this sort of small print. So it's evolving. There's an insufficient knowledge base. It's certainly getting better, but there are certain obvious things that need to change. And one of the old-fashioned nomenclature is very confusing to clinicians. That's something that we sort of have to think about. The science is evolving what I call across the integrative spectrum, right? The investigative spectrum. So both at the level of CNVs, and the issue there is penetrance. Right, the estimates of penetrance are changing as we study broader and broader populations, as well as things such as annotation of variants, and how complete is the annotation for certain things such as pharmacogenetics. So there's a lot of data out there, right? But this data is complex, mostly is of modest effect, admittedly. And so that requires a lot of balanced reasoning. And this isn't even mentioning about the problems of implementation. So you get a test, where is the result? For us, you get a piece of paper. If you have, if somebody does due diligence, they'll scan it into the EMR, but it'll be a PDF and it will almost never be seen again, right? So we have to think about how to actually show the results and show the results for the patient across health systems and across different physicians and different doctors. It can't just be a one and done sort of deal with genetic testing. How are we doing with our education? N of one. This isn't working, but just to say, here at Johns Hopkins, we only have a single session on precision medicine. It's taught by pharmacologists. The slides were excellent. I saw them. They were very, very good. But we don't teach it at all in psychiatry, in the preclinical, and we don't teach it during the residency, right? So we're asking a lot for clinicians, given that we're actually not spending enough time teaching them. And finally, so what are some of the thoughts about how we need to sort of change? Well, psychiatric genetic testing is actually here. I think most of us would say it's not really ready for prime time, but in a sense, tough. It's here, it's with us. So our educational mission needs to step up its game. And I think we really need to be part of the genetics education more integratively, right? I don't like seeing psychiatry teaching things differently than the rest of medicine, because this is very complicated. So we need to learn about genetics, bioinformatics, predictive modeling, not everything, but we've got to touch on some of that. That's too much for just us to teach. And it's important to the whole field of precision medicine. So it really needs to be interdisciplinary, as you've heard from other speakers. And again, we can then come in, those of us that are interested in the psychiatric genetics, come in and sort of augment and provide additional sort of context-specific information. So thank you. I think I'm just at my 10 minutes. Happy to talk more about this in the chat session. I'll pass on to chat.
Thanks, Fernando. I'm sure there'll be plenty to talk about in the chat session on that subject. And you touched upon pharmacogenomics, and our last talk of this session will be led by Chad Bousman, who will go into further detail on pharmacogenomic testing and recommendations for clinical psychiatry. Chad?
So, sorry about that. So in the brief time that I have here, I want to provide a summary of the review and consensus on pharmacogenomic testing in psychiatry that was conducted by the Pharmacogenomic Testing Subcommittee, co-led by Daniel Mueller and myself, and with significant contribution from the 29 members that are listed here on the slide. So I don't think it's controversial to state at this point that medication efficacy and tolerability vary from person to person. and that this variation can in part be explained by pharmacogenomic factors. And this notion has spurred a increase in pharmacogenetic testing options worldwide. But despite this availability of testing options, the adoption of pharmacogenetic testing in psychiatry has been quite modest. And some of this has already been touched on in the education session and Fernando of why this is, and it has a lot to do with the diverging perceptions of the evidence base, variable knowledge among psychiatrists about pharmacogenomics, and mixed views on the clinical utility of pharmacogenetic testing. So recognizing this and recognizing that there's no consensus within the field on the use of pharmacogenetic testing in psychiatry, we sought to do an in-depth review of the literature and develop some recommendations. So this is just a brief overview of our review and consensus process, which is shown here in the figure. The first step was goal setting. And so we had three main goals for the subcommittee. The first was to review the pharmacogenomic evidence. And this included the published literature, prescribing guidelines, as well as the product labels or drug labels related to psychotropic medications. We then wanted to highlight key considerations and limitations to the use of pharmacogenetic testing in psychiatry, because we do realize that just having the evidence is not enough. You need to really jump that gap into the implementation. And then finally, offer recommendations for the use of pharmacogenetic testing in psychiatry. So as you can see here in the figure, it was an iterative process where we had evidence discussions, both virtual and in person. We had two in-person meetings through the WCPG annual meetings in Glasgow and most recently in Anaheim. From those discussions, recommendations were proposed to the full group. And when concerns were raised, we either went back to discussions or most of the time, it was just modifications to those recommendations until consensus was achieved. So for our first goal, when we looked at the current pharmacogenetic evidence base, what was clear was there's 42 gene drug pairs with actionable pharmacogenetic based guidelines. And this comprises 7 genes and 34 drugs. And those are depicted in the figure here on the left. And as I think you can appreciate, CYP2D6 and C19 are the main players, having 35 of the 42 gene drug pairs associated with one or both of those genes. And antidepressants and antipsychotics make up the majority of the actionable gene drug pairs that have dosing guidelines. So as I mentioned before, having evidence is only part of the equation. There are a number of considerations and limitations to getting pharmacogenetic testing into practice. And here on the right is just a figure of the different components of, or different potential limitations or considerations that we looked at. I don't have time in this talk to go through each one of them in detail, but I will highlight a bit for each. So on the front of selecting and ordering tests, it was clear that there are many options available. In the US alone, there's more than 75 laboratories that offer pharmacogenomic testing. However, there's no evidence to suggest that any one laboratory test is superior to all the others that are out there. From a content validity perspective, it's worth noting that there are no regulatory standards at the moment for test content, and that leads to a variety of different test formats and what alleles are being tested and what genes are being tested. Again, this has been alluded in other talks as well as in the Education Day pharmacogenetic talks. Analytical validity is often overlooked and taken for granted, as it's just, as long as it's an accredited lab, analytical validity is a check. But we do know that even within accredited, between accredited labs, analytical validity can vary. And this is particularly the case for CYP2D6 with the complex structural variations that make genotyping quite tricky. For clinical efficacy, there's been 2 recent meta-analyses of RCTs that support the efficacy of pharmacogenetic testing in major depressive disorder. But other indications, bipolar, schizophrenia, and anxiety require more studies. From the feasibility standpoint, there's quite a bit of information on this in the literature. Turnaround times being the big issue, particularly for preemptive testing. Turnaround times can range from 2 days to 3 weeks, which is obviously too long in some care settings, particularly acute care settings. And the cost and reimbursement barriers are well known as well. Cost effectiveness, most of the evaluations that are out there suggest pharmacogenetic testing is cost effective or cost savings. But it is worth noting that in psychiatry, these tests or these evaluations have been done by commercial interests. So we do need some independent cost effectiveness analyses. But I can say that the current evaluations are promising. And then finally, the interpretation and delivery. This is actually quite a large category, but we wanted to emphasize that, and as others have has emphasized as well, non-genetic factors can play an important role in the interpretation of pharmacogenetic test results. But the laboratories that offer pharmacogenetic testing often do not account for these factors. So it really is on the healthcare provider to merge those different data points together before making a decision. I'll finish off with our consensus recommendations. And we really have an overarching statement about pharmacogenetic testing. It's a tool to enhance rather than offer an alternative to standard treatment protocols or other personalized prescribing strategies. So this is not a replacement. There's a number of personalized prescribing strategies that have been used for decades, and this is just another tool in that toolbox for clinicians. So our overarching recommendation was that the current evidence supports pharmacogenetic testing for specific antidepressants, antipsychotics, mood stabilizers, and ADHD medications, but it does not support testing for anxiolytics, addiction medications, genetic variants, and pharmacodynamic genes, particularly serotonin transporter COMT and THFR. These are genes that are often offered on panels, commercial panels, but currently there's just not the evidence base to support that. I'll end there. If you have any additional questions, happy to talk about those in the breakout session, as well as we'll point you to the paper that discusses this in more detail that was just accepted for publication in pharmacopsychiatry, which should be online in the next couple of weeks. Thank you.
Thanks, Chad. And thanks to all of our speakers for, I think, we're a very stimulating set of talks. We now invite you all to join our speakers and other committee members in the chat rooms, which will continue until 1:30. The links for that can be found on your online agenda.